since our next topic is the reproductive system, I want to go over some stuff about how cells and organisms reproduce. This is going to be background information for some of you. If you've had the Cells and Genes course, then you've probably had this material before. But it's good to review it, and some of you may not have covered it in your original intro biology class. This is basically what I call my making copies lecture. And first off, let's get started with some terminology. First term that you need to know is about DNA. DNA is the molecule that replicates. And in eukaryotes, like us and pretty much all other animals and plants and most things that you've ever seen, that DNA is contained in chromatin. Chromatin is a combination of DNA and protein bound together in an organized fashion found in the nucleus of a cell. The DNA is in the form of a DNA molecule, which is a double-stranded double helix. So it's two nucleotide polymers that are bound together with hydrogen bonds. A single DNA molecule and all of the proteins that go into making it chromatin, which are mostly proteins that are called histones, which are organizational proteins that the DNA is wrapped around, that's called a chromatid, right? So a chromatid is all of the DNA and all of the proteins in one molecule of DNA. A chromatid contains a centromere somewhere in the center, and on either side of the centromere are what are called arms, and at the end of the arms are telomeres. Telo means ends. So chromatids are made from chromatin. A chromatid and a chromosome are very similar in many ways, and sometimes a chromatid is a chromosome. But the chromosome is a unit of chromatin that moves and acts as a single entity. Because your cells have to double their DNA, they have to replicate their DNA before they can divide the cell, sometimes a chromosome will contain one chromatid, and sometimes a chromosome will contain two chromatids linked together at the centromere. If a chromosome is made of two chromatids bound together, those two chromatids are called sister chromatids. This is an example of a chromosome, and this chromosome has two chromatids. You can see this up here. This is one chromatid here. This is one chromatid here. And they are bound together at their centromere. This is all one chromosome because if I were to, say, tug on one arm of this, the whole thing would move together. It's all bound together. However, as we'll see later on, if I were to clip this binding, like cut the binding here at the centromere, and these two chromatids would float away from each other, now they would be two chromosomes because they're no longer bound together. The life cycle of a cell is what's called the cell cycle. This goes from its birth all the way until it divides or dies. So it's the life of a cell from its time it's first formed until its own division into two what are called daughter cells. The cell cycle consists of two broad phases. The longest is interphase. Interphase is all of the stuff that happens when a cell is not actually dividing. The other phase is mitosis, or M prophase. This is what happens when the cell is dividing. Most cells spend most of their life in interphase. Interphase has 3 subphases in it, what are called G1, S, and G2. The G technically stands for gap, so this is the first gap phase, this is the second gap phase, but you can also sort of think of it standing for growth, right? At the start of a cell's life, it has just divided. It's small, it's young, it's fresh. It needs to grow before it can divide. And so during G1, it is mostly just growing. During G1, the chromosomes are 1 chromatid each. The DNA has not yet divided. In the second phase, S phase, S stands for synthesis. In this phase, the cell decides, all right, I'm going to replicate myself eventually, and if I'm going to do that, I'm going to need to make a copy of my DNA. That is the primary thing that's happening during S phase. It's taking all of the DNA, all of the chromatids that it has, and it's making a copy of each of them. So the amount of DNA that's present in the cell will double during S phase. However, the number of chromosomes stays the same. That's because this new DNA that's been replicated, remains bound to the original chromosome at the centromere. So here, after S phase, after the DNA has been replicated, the chromosomes have 2 chromatids. They're that standard X-looking chromosome that you've probably seen many times. After the DNA is replicated, you enter into the second gap phase. This is where the cell is ready to reproduce, right? It can't reproduce until after it's doubled its DNA. Now it is in the second gap phase. It's basically waiting for a signal that tells it, all right, you can go ahead and reproduce. When it gets that signal, it enters into mitosis. Mitosis consists of, or the mitotic phase, M phase, consists of 2 parts. Mitosis, which is where the chromosomes will split and migrate to opposite sides of the cell, and cytokinesis. Cyto means cell, kine means movement. This is where the cell literally splits in half, and 1 half will contain half of the DNA, the other half will contain the other half of the DNA. Mitosis has 4 or 5 phases, depending upon which textbook you look at. I'm going to describe mitosis in terms of 5 phases, which are Prophase, Prometaphase, Metaphase, Anaphase, and Telophase. It should be noted that some textbooks omit Prometaphase and just divide the events into either prophase or metaphase, but that's not really an important distinction. Everyone agrees on what happens during mitosis. The only question is how we want to divide that up and talk about it. This is how I'm going to talk about it. So this is a cell, all right? This is a cell before mitosis. It has some important cellular features. We've got a nucleus. That nucleus contains uncondensed chromatin. So this is chromatin, DNA plus protein, that's unwound so that it's accessible. And in this state, you can't actually see individual chromosomes with a microscope. It's basically all unpacked and spread all over the place. We also have cellular organelles, like the endoplasmic reticulum here, the Golgi body, and mitochondria, as well as a number of other organelles as well. In animal cells, which is what we're concerned with, we also have two specialized structures that are called centrioles. Centrioles make a part of the cytoskeleton, which is the protein network that gives structure to the cell. The part of the cytoskeleton that they make are called microtubules. They're going to be very important to the process of mitosis. So this is the cell during interphase, during its normal stuff that it's doing, right? Now let's say the cell begins mitosis, it gets the clearance, it's going to start dividing. Well, what happens? During prophase, one of the first things that happens is the chromosomes condense, right? They wind themselves up really, really tightly, and they turn in to those tightly wound chromatids bound together at centromeres, like the picture that I showed you earlier. And so now, at this point, you can actually see the discrete units of chromosomes. In addition, the centrioles are going to start migrating to opposite sides of the cell. During prometaphase, most of the cell's internal organelles will actually disappear. In order for the cell to divide, these chromosomes need to be able to move around. And so that means that the nucleus is actually going to be taken apart, and same with the Golgi and the endoplasmic reticulum. Those are all going to be basically broken apart and packaged into little storage vesicles that will go one way or the other. Interestingly, this doesn't happen with the mitochondria. But the mitochondria will move to one side or the other so that some mitochondria end up in each half of the cell. The other thing that happens in prometaphase is that the spindle apparatus starts to be formed. The spindle apparatus is this network of microtubules that comes from the centrioles, or centrosomes. And some of these microtubules will actually spread across the cell and join up with other microtubules, but a fair number of them will actually grab onto the centromere of the chromosomes. During metaphase, what happens is that all of the chromosomes of the cell line up in single file down what's called the midplate or midline of the cell. So here you can see all of these chromosomes are lined up single file down the middle of the cell. This is the key signifier of metaphase. In anaphase, the first thing that happens is that the centromere is actually split apart in these cells, or in these chromosomes. So that now what used to be one chromosome with two chromatids becomes two chromosomes with one chromatid. And those chromosomes will walk themselves down the line of the spindle apparatus. Just like you're sort of reeling in a fish. The other thing that has happened is these unattached spindles have actually pushed against each other in order to elongate the cell. And now you can see that half of each chromosome has been put on each side of the cell. This is the sister chromatid of that. This is the sister chromatid of that. Since DNA copying is, barring mutation, an exact process, these are typically considered to be identical chromosomes. Sister chromatids are identical to one another, or at least usually identical to one another. Mutation is always possible. The last phase of mitosis is telophase. Remember we said that telomeres were the end of the chromosome? Well, telophase is the end of mitosis. And in telophase, what happens is we start to put the cell back together. So we're going to form a new nucleus around each of these clusters of chromosomes on opposite sides of the cell, and we will begin to reestablish the cell's internal organelles. The other thing that happens during telophase is cytokinesis. So the cell will actually physically divide. There's actually a ring of proteins that works like a belt to tighten around the middle of the cell and cut the two halves of the cell off from one another. At this point, the chromatin, or the chromosomes, uncondense, go back into being uncondensed chromatin, and these two cells separate and begin the cell cycle again. Obviously, this is one of the most controlled events in any cell's life. We have a term for cells that replicate without control, and that's cancer, and it's usually a bad thing. So controlling when cells replicate and when they don't replicate is one of the most fundamental and important processes that can happen. So there are control points in the cell cycle. And in most animal cells, there are three main control points. You see this arrow here? This is pointing to where in the cell cycle we are. So we're starting off with a fresh cell. It just divided. We have a new daughter cell right from a mitotic split, and it's going to go through G1. The checkpoint at the end of G1, right? So at the end of G1, one of two things happens. Either there is an external signal, a hormone, like growth hormone or something like that, that says, all right, you are cleared to reproduce. You are allowed to divide. Or there's no external signal. If the external signal is received, if growth hormone is received, then the cell will go on into S phase and start replicating its DNA. Replicating DNA is very energetically expensive, so you don't want to do it unless you're actually going to divide the cell. If this cell isn't going to divide, it's just going to be, I don't know, a muscle cell or a nerve cell or one of your skin cells that's going to eventually go all the way up through the layers of your epidermis and eventually flake off without dividing. There's no point in doing that. And so it will then go into a phase called G0 or G0, which is basically it just stops there and continues being a cell, but it's never going to reproduce. It's never going to divide. It's just going to continue doing its cell thing. But if this cell does get the external signal, the hormone, to reproduce, then it passes the G1 checkpoint and moves on to the next checkpoint. Like it goes through S phase, doubles its DNA, goes through G2, and at the end of G2, there's another checkpoint. This is controlled by internal signals, specifically an internal protein called cyclin, which is the internal protein that controls the cell cycle. What this does is it basically ensures that the cell has spent enough time going through S and G2 phase that it didn't rush itself along and didn't make lots of mistakes. It makes sure that the cell has enough time to replicate its DNA and, crucially, to check its replication to make sure that it didn't make any mistakes. Mutations happen actually fairly frequently, but most mutations the cell will actually catch, go back, and fix. This isn't to say that mutations can't happen. They do happen all the time. But in human and animal cells, only about 1 out of every million or 10 million bases in your DNA actually gets a mutation that gets through all of these problems. So we want to make sure that we've spent enough time on our work checking everything, right? Once you pass the G2 checkpoint, because cyclin is like made slowly and it builds up over time, and then when you get enough of it, you can pass through this checkpoint. Then you move into mitosis. And that is where the third checkpoint is. This happens during, I know that says interphase, but this actually happens during metaphase. And what it does is it checks to make sure that the spindle apparatus is properly set up, that all of the chromosomes are in their proper place, and that you have the right number of everything. If the cell spindle apparatus doesn't get set up properly, it can cause major problems for the cell after division. It might end up with too many or too few chromosomes. And those are problems that can eventually result in cancer. So during this checkpoint, what it does is it checks to make sure that mitosis is proceeding properly. Everything is hooked up right. If things aren't hooked up right and it spends too much time and can't get itself figured out, the cell will actually die. It will trigger into what's called apoptosis, which is programmed cell death. But assuming that everything is working properly, the spindle apparatus is set up right, then the cell gets clearance to continue with mitosis and cytokinesis and divides. A little bit more terminology, because we've talked about how cells divide, and so that's how you get new cells. But we also know that when people make new people, it isn't just a matter of splitting in half. You don't just make, a bunch of copies of yourselves and grow a little copy of you. No, humans and most animals and most eukaryotes reproduce through sex. And with sex, you are going to pass on part of your DNA and combine that part of your DNA with another person's part of their DNA. We have some terms for these type of cells. Haploid means that you contain one set of DNA. Diploid means that you contain two sets of DNA. Humans are diploid creatures. You have two copies of every gene in your body. You got one of those copies from your mom, and you got one of those copies from your dad. That's what makes you diploid. You have two copies of each DNA strand of each gene. When you pass on your genes to your children, you will only pass on half of your DNA. You will make what is called a gamete. These are sperm or eggs. Each gamete that you make will only contain one of each gene, one of each chromosome. So normally you have two chromosome 1s, two chromosome 2s, two chromosome 3s, all the way up, right? The gamete, the sperm or egg that you produce, is only going to have one chromosome 1, one chromosome 2, yada, yada, yada. These paired chromosomes, like you have two chromosome 1s, those are called homologous chromosomes. Homologous means similar, but not identical. Remember, sister chromatids are supposed to be exact copies of one another. That's not what a homologous chromosome is. Homologous chromosomes contain the same genes, but they might have slightly different versions of each gene. So, for instance, if one chromosome has genes for hair color and eye color and nose shape, then the other homologous chromosome will also have genes for hair color, eye color, and nose shape. However, that first chromosome might have genes for blonde hair, green eyes, and a wide nose, whereas the second chromosome might have genes for brown hair, blue eyes, and a narrow nose. So the genes are the same, but the version, the specific type of that gene, what's called an allele of that gene, may be different. Your body produces these gametes, haploid reproductive cells, each of which contains one copy of each homologous chromosome pair. But it's the chromosomes do what we call independent segregation. That means that you don't pass on all of the genes that you got from your mom or all of the genes that you got from your dad. It's random. You might pass on one chromosome that you got from mom, chromosome 2 that you got from dad, chromosome 3 that you got from dad, chromosome 4 that you got from mom. It's random which ones you pass on. When two gametes unite, when sperm and egg fertilize, what you've made is a zygote. A zygote is a diploid cell formed by the union of two gametes. Last thing, you have more than one type of chromosome. In fact, humans have 23 chromosome pairs. So you have 2 chromosome 1, 2 chromosome 2, 2 chromosome 3, all the way up to chromosome 22. Each of those is what's called an autosome. With autosomes, the paired chromosomes are homologous. They'll have the same genes on them. But humans and most mammals have a special chromosome pair called the sex chromosome. The sex chromosome is paired, but the two types of chromosomes aren't exactly the same. Humans have X chromosomes and Y chromosomes. If you have 2 X chromosomes, typically your body develops female, although we'll see later that there are some things that can interfere with that. If you have an X and a Y chromosome, typically your body develops male, although again, there are some things that can affect that. The X and Y chromosome are not homologous to each other, although 2 X chromosomes would be homologous to each other. The process by which you take a normal cell, a diploid cell, and make a haploid gamete from it is called meiosis. Not mitosis, meiosis. And it's a very important process. Even though it's done a lot less than mitosis is, this is absolutely essential for sexually reproducing organisms. If you did not undergo meiosis, then when I passed on my genes with, somebody else, when we had sex and had a kid, it would end up having twice as much DNA as we did. And then its kid, our grandkid, would have 4 times as much, and then 8 times as much. And that screws things up royally. So diploid organisms that reproduce through sex have to cut the number of chromosomes that they pass on in half before they pass on their genes. This is called chromosome reduction at meiosis. The whole point of meiosis is to make gametes that contain one copy, haploid, number of DNA. The steps of mitosis and meiosis are similar, but there are a few differences. First off, while they use the same phases, prophase, metaphase, anaphase, and telophase, mitosis goes through this set once and done. They're one and done. Meiosis will actually go through these steps twice, with some slight variations between them. So if I say this cell is in metaphase, you know that it's going through mitosis. If I say that it's in metaphase 1, it would be in meiosis. And in meiosis, the steps are named by which half you're in. So it would be prophase 1, metaphase 1, anaphase 1, telophase 1, then prophase 2, metaphase 2, anaphase 2, telophase 2. If it's got a number after it, you're talking about meiosis. Mitosis produces two genetically identical cells. Meiosis, since it's going through the process twice, is going to produce 4 cells. And since the chromosomes are getting randomly assigned, each one's going to end up with half. What you actually get is 4 non-identical cells. Each one will have a different combination of chromosomes. Like the first cell might have chromosome 1, 2, and 4 from dad, but 3 from mom. You know, the second cell might have 1 and 2 from mom, but 3 and 4 from dad, that sort of thing. And in fact, the next thing is that you don't actually get a whole chromosome from mom or a whole chromosome from dad. During the first bit of prophase, prophase 1 in meiosis, homologous chromosomes swap DNA with one another in a process that's called crossing over or homologous recombination. Basically, what this means is that you don't pass on an entire chromosome that's the chromosome you got from mom. Any chromosome you pass on is going to be a mishmash. It's got some of the chromosome you got from mom and some of the chromosome you got from dad, all kind of patchily mixed together in it. This increases genetic diversity. The last difference is in metaphase 1 of meiosis. And we'll see this. Remember in metaphase of mitosis, how the chromosomes lined up single file down the center of the cell? Well, in metaphase 1, as we'll see, of meiosis, they're going to line up 2 by 2. So let's take a look at our cell before meiosis. This is the same cell. I use the same picture, right? It's got the same stuff in it. You got a nucleus, you got some organelles, you got some mitochondria, you've got your centrioles, or centrosomes. In prophase 1, same sort of steps occur, actually. Your centrosomes are going to migrate to opposite sides of the cell, and your chromatin will condense into discrete chromosomes. Now, note that I've got 2 different colors of chromosomes here. I'm using red to represent the paternal chromosomes, the ones that you got from dad, and blue to represent the maternal chromosomes, the ones that you got from mom. So they're going to pair up. This organism that I'm showing you here has 6 chromosomes in 3 chromosome pairs. Humans have 46 chromosomes in 23 chromosome pairs, but that's really more X's than I want to draw in this PowerPoint. In metaphase 1, the internal organelles, the nucleus, endoplasmic reticulum, and stuff like that have all been dissolved and packed away, and we can see that the chromosomes line up 2 by 2 down the midplate, and the spindle apparatus, each spindle is going to grab onto one of the homologous chromosomes. These are lined up paired. by the way. So this top one, that's chromosome 1. This is daddy chromosome 1, this is mommy chromosome 1, but they're both a homologous pair of chromosome 1, so they have the same genes. Note that the lining up is random. You don't have all red down one side and all blue down the other. During Anaphase 1, you're going to separate the homologous chromosome pairs. This is where chromosome reduction takes place. So before, we had 6 chromosomes in 3 pairs. Now we have 3 unpaired chromosomes on each side. And again, the spindle apparatus will elongate the cell. In telophase 1, the nucleus reasserts itself and the cells divide. With meiosis, we're not done. We now go into prophase 2. What happens in prophase 2 is pretty much the same as what happens in prophase 1. Chromosomes recondense, and the centrioles spread to, or centrosomes spread to opposite sides of the cell. In metaphase 2, the spindle apparatus forms, and metaphase 2, like metaphase of mitosis, the chromosomes are going to line up single file down the center of the cell. In anaphase 2, we're going to split the centromere of each of these chromosomes, and one sister chromatid will head each direction. And in telophase 2, we are left with 4 haploid cells, each of which has a different set of haploid genes. These are cells that will go on to become gametes, or sex cells. When we're talking about the reproductive system, I will often talk about what phase of mitosis or what part of mitosis is going on during each part while, describing the process of making sperm or making eggs. Because, of course, this is the simplified version, and in reality, it gets a little bit more complicated than this. But remember that cells are haploid after they have completed meiosis 1. That's a key determining point in the process of gametogenesis or gamete generation.